we're merging into the panel, if you hadn't noticed. I'm going to sort of make a couple of quick comments, and I think everyone behind me is going to nod their heads who's seen SLC6A1 patients. Yes, sleep challenges are real. There is a dysregulation of the ability of the brain to go through normal sleep-wake cycles, to maintain appropriate sleep transitions. It's challenging. There are medications that can help. They don't always help all the way, but it's worth trying. And so we should have those conversations and we should try things. We saw nice regression data. Regression is real. I think we have questions still about regression. How often is it an autism regression? How often is it associated with epilepsy onset? How well do patients eventually recover or not recover from those elements of regression? And do they typically happen mostly in the younger ages, or do we get random regressions even at older ages? I think those are some outstanding questions that we need a little bit better, again, prospective natural history data to help us answer in really well-controlled and rigorous ways. And I don't know, the behavior side of it, we've been talking about it all day. And my biggest conclusion is that you can't have comprehensive care for a patient with SLC6A1 without having availability of behavioral health as part of that. That's it. Whether that's developmental pediatrics or psychiatry or whoever it is a fundamental component of being able to treat SLC6A1 patients. We all have an obligation to make sure that that's available to our patients, and that's it.
Thanks. So my name is Anup Patel, and I'll have everyone introduce themselves. Oh, I get to work at Nationwide Children's Hospital with this wonderful person, Dr. De Los Reyes, and Allison, who you heard from earlier. Before I turn it over to everyone, I've gotten to work with a lot of these folks that are on the panel, and it's been a true honor to be a colleague and be able to work and collaborate with all of you. You guys are amazing with what you do, and your approaches are just awesome, and it's just inspiring. But then to Allison, we're going to talk about why gene therapy takes so long. But when you heard that story, she moved heaven and earth in a pandemic to get this to happen. And so if she can do that, then we can do that too. And so kudos to her and her work, and to Emily for being such a strong clinical advocate. So I want to give you two a huge round of applause again. So I'm going to pass the mic on and have everyone introduce themselves and what institution they represent, and then we're going to dive into it.
I'm Dr. Marie Barnett. I'm coming from the University of Colorado. I specialize in neurodevelopment.
Pam Bojamovsky-McDonnell. As you heard earlier, I do epilepsy and neurogenetics at the Children's Hospital of Philadelphia.
I'm Emily De Los Reyes. I am a neurologist, a pediatric neurodevelopmental physician, and at this era, I'm doing a lot of rare disorders.
Zach Gernspan at Weill Cornell Medicine. I'm a pediatric epilepsy physician.
Scott Demarest, and I do whatever Amber tells me to do.
That's a good way to approach life. All right, so we're going to do 3 questions today, and I think a lot of what we're going to cover has been discussed throughout the morning and early afternoon session, but I think this is going to be a nice panel to summarize some ideas. The 3 questions, I'll tell you what they are, and for those that have worked with me, they'll understand why I have question number 3 as I listed in there. So why does gene therapy for SLC6A1 take so long? We've heard some of those. I want to hear from you guys. What could we do to accelerate that development for these gene therapy products? And more importantly, number 3, which is the part where you guys would expect, what are we going to do together in the next 12 months to make this happen? And that's the timeline we have. So when we come back a year from now, we will have some of those answers, but that's the timeline we're going to try to figure out. So let's start with number 1. Emily, I'm going to start it with you on, you've been so instrumental in this journey of gene therapy development. Why does it generally take long? And you've had a lot of experience in that. Zach has a mic.
I'm looking at Dr. Bradbury there. We worked on the, and I think a fair amount of that, that's what we saw was the toxicity, right, Amber? In the knockout model, in the Maxwell mice. And we just wanted to make sure that we had the correct dose. I think somebody talked about haploinsufficiency, and so we knew that he still had the gene, he recognizes the gene, but we don't want to push them over the edge. I think you heard them over and over again. Our children, I know, I hear, and I think I learned a lot today. I'm a newbie here, so I love going to conferences, but because I hear from the families. And so I think that's the main thing, is we wanted to make sure the dose was correct, that the toxicity was, that the reasons for toxicity was correct, because our research and clinical grade vectors were like a little bit different, and we wanted to make sure that we were going to the right way and we did not harm Max.
Anyone else want to comment on that?
I can, you know, so Susan and I know each other through a different rare disorder, IRF2BPL, and we've been texting with the mom of that disorder during this conference. So I wanted to reflect on what Dr. De Los Reyes said about this worry for overdose. But I want to reflect on IRF2BPL because it was really fast. This was a child with very severe developmental and epileptic encephalopathy, not really rolling over, horrible hypsarrhythmia, horrible seizures all the time. We found a gene, this ultra rare IRF2BPL. The family, we made the diagnosis in February of 2024. The family made a decision to go for an AAV9 therapy that June. And the next April, we did a cisterna magnet injection with an AAV9 gene therapy. Susan deserves a huge amount of credit for that because of the manufacturing challenges. So these can go fast. And I think for SLC6A1, you know, when you talk to the FDA, like for that disorder, for other ones we're working on, you can make these arguments asking for regulatory flexibility because of the severity of those illnesses. But with SLC6A1, it's you know, the regression is there, but it's not degenerative. You don't get the sense that children are ticking time bombs, if I can be informal. And so I think you have to be cautious. You don't really want to hurt anybody, and so the caution implicit in this approach feels very wise to me.
I think of it in sort of 4 buckets. I think the limitations for any gene therapy, but funding, of course. I don't know if you can hear me. There's what I put in the technical bucket, which is everything that you heard about this morning: dosing, safety, making things, getting mice, getting AAV, all of that. Access. So we all come from big centers, but what happens to everyone in the middle or in other countries, and how do we get everyone diagnosed properly? And then the last one, which you just alluded to, is the broad spectrum of the disease. So we have parents that were diagnosing as their children get diagnosed because they sort of were higher end or never had seizures or seizures stopped. And then we have very severely affected children who are being institutionalized. And with that really broad spectrum, it's hard to develop sometimes those clinical outcomes that you're saying, we're counting seizures because we love to count seizures as epileptologists, but not the most important thing always. And so in my mind, it's, if I put buckets in there, then we can come up with solutions for that. And so that's how I think of it for, you know, any genetic disorder.
Yeah, and speaking of that, and Marie, I'm going to turn this to you. You know, you have had a lot of experience with getting people from different portions of the country together. Can we interrupt you for a moment? Yeah.
Sorry, I'm going to add a comment. So my son Charlie was in that behavioral video, and just a comment to the ticking time bomb statement. In a lot of ways, a lot of our kids might not feel like ticking time bombs. Charlie's a ticking time bomb. Every minute of every day, he is a risk to hurt someone in my family, someone at school, or himself. And he's 8 years old. He is on his 4th school for kindergarten. He was locked in a closet for an hour for behavioral challenges. Obviously, that's illegal and not okay to do, so I don't recommend that for anyone. But I think sometimes that's where, as parents, the challenge lies. Obviously, we don't want you to kill our kids, but I would rather take that risk than live a life of knowing that I allowed him to hurt someone, or being in fear for my daughter, or a stranger, or himself, or, I mean, I don't know. We're at risk of something worse than death quite honestly.
Yeah, and this is something I just hear a lot from the families is we're always in a catch-22 because we're not classified as ticking time bombs, meaning death isn't imminent in 5 years. But if you knew when they were 5 years old that they were going to live under a bridge or be a school shooter or something, I mean, against all of these interventions we're trying, I think a lot of us would probably choose the terminal option than to see this go into adulthood.
Yeah.
To Mike.
So we got Liam when he was almost 3, and his sister was 5 weeks old. Their birth mom was a heroin addict, and Anna Grace was addicted to heroin, so she was in the NICU. They were taken from us after 2 years, put back with birth mom, and suffered a lot of abuse. Liam saw Anna Grace get assaulted and abused. Fast forward to Liam being 15, Anna Grace is 12. Liam has been in treatment for a year at Cholita in Wisconsin. He had deep therapy there. We got him back home in June. In September, Liam had been threatening to kill us all again in detail, cutting Dad's eyes out. This has been going on for years. Since he was, they removed him from regular school, put him in a school outside of the school. They made me bring him home because he was so violent. So I had to supervise homeschooling online. So in September, Liam had been threatening to kill us all and anybody else, and in detail, cutting Dad up, putting him down the garbage disposal. So that morning, Liam had threatened us again, and we were trying to keep him inside so that he wasn't down by the school bus causing problems and all the neighbor kids seeing it. They're very young. And my husband tried to hold him in the house. Liam's very big. Scott's very small. And Liam went out the door. Scott tried to stop him. Liam shoved his body backwards onto Dad, and they fell backwards, and Liam's grinding his elbow into Dad. I'm trying to get him off. My husband said, "I can't breathe." And Liam put his hand up by my mouth, and I ended up biting him. And then he did it again, punched at me, so I bit him again. We've had multiple police interaction at our house. Well, my 18-year-old daughter called 911, and she asked Mom, should I call? And I said, yes, please. And they came. Liam was removed from our care for his safety. We've been locking up knives for years. My daughters have been sleeping with me. My husband's been sleeping downstairs. Liam will kill somebody someday. He will. We've been prisoners in our own home for years. Nobody's listening to us. Nobody's helping us. So he's currently in foster care, and we're facing charges.
Liam's in foster care and they're facing charges. And I think what we're all trying to balance is how do you balance not doing harm, that they're generally stable. You don't want to hurt them because they're not terminal, but maybe it would be better if they are, if this is the alternative.
Yeah, Scott, do you want us to take that?
Good luck, Scott.
So just to be very clear, and this has actually been proven out, the fact that Maxwell was treated under an N of 1 IND means that he met the definition that it was either life-threatening or severely debilitating. That's the terminology from the FDA. And I don't think there's any doubt that we're talking about that with SLC6A1. We've been talking about the behavioral component of this as one of the sources because it's not a challenge for every single family. But for those that it is, it can be an incredibly severe and debilitating challenge. And we absolutely meet that definition as a community. So I don't think that the question is whether or not this is a severe enough condition for us to be able to take heroic action to try to address it. That may or may not be right for every family, and that is okay. And every family should have the option and ability to make that choice when it's right for them.
Yeah, and I think, if I could weigh in on the answer to the question Amber poses, which I think is an important one, and, this is going to sound so boxy, and again, for those that know me, won't be surprised. But I think we haven't worked together enough, and so we need to be working closer together and learn. Maybe the dose is low, maybe not, but maybe we can develop a better, vehicle that that will help us get up to a better dose or a better, absorption or whatever you want to call it rate. Uptake rate, sorry, that's the word I'm looking for. But I don't think that's going to happen in silos. And that's where I think it's important that we have come together and try to continue to do that. Zach, you want to say something?
We're spending a lot of time talking about whether it's a low dose or not. Like, we dosed, guys.
Right.
Like, what a luxury to have to worry about whether or not it was the right dose and be able to, come on. This is a huge milestone.
Just a quick comment. I think these arguments and these sort of situations, that the FDA guidance for disorders that are severe or life-threatening specifically talks about regulatory flexibility, and so things can move fast. I think, Pam, you put it well, that like you have to have lots of things line up to move fast, and then families are willing to take on risk. There's a diversity of how comfortable families are with risk, and when you have an N of 1 and you have a family that's willing to take risk, it's a different story. And then the tension becomes N of 1 versus N of community, and how do you find that balance? So I think what's been tremendously exciting for me is just the number of therapies that are in development, and so there's multiple options and multiple things moving forward.
I want to encourage the families that N of 1 is extremely important because so far it's safe. And I have used this cassette for other diseases, and I'm seeing the same safety signals, so that's good. He's not the first one with that cassette, so that's always good, but he's the first one with the disease, so I had to figure that out. Number one. Number two, we are moving towards other mutations, so I also, that's very encouraging. And so like what you guys said, it takes a community, it takes a village. And so that's a village of sharing our mice or pigs or whatever. So I think that's good. And then number three, the dose. I know manufacturing is different, titration is different. So when Allison can talk about this, that, it's not necessarily just the dose, but at the same time, after the Rett's death, there was a child who died of Rett syndrome with intraventricular therapy, complete meltdown. And so now we're backing off because people were like pushing the dose. And I'm like this kind of person who's again, every night when we have a new patient, we dose. We all don't sleep. We are on PPIs, we're on medications just so that, oh my God, I dose another child. And not just one child, it's every child that we dose. And that's anywhere from an SMA child to Duchenne's to whatever. So it's not easy, guys. But at the same time, I think that our foot is in the door. We know it's safe. I know, for example, I use steroids for immune mediation, and that's also very challenging, guys. So for the families who have behavioral difficulties, that is also, again, this is gonna be a basket study. So kids with behavior are treated with different immune versus steroids or whatever. So there is a little bit of going back to the drawing board again. But I just want to encourage the families that the signals so far are very good. And thank you. I told Amber today, you're the sweetest families. I've presented gene therapy before, and this is the first time I've been honored this way. So you're the most wonderful families I've met. So in my life.
That's really sweet. All right, Marie, I would like to get your comments. We talked about, why some of the challenges of why it takes so long, but, what are some opportunities that can accelerate development of these products? And I'm turning it to you because you've done a really nice job shepherding kids from all over the United States with getting the natural history studies together. So anything that you've learned that we as a community can learn as we start to develop or want to develop these therapies?
Yeah, so one thing that's come up a lot when considering who's going to participate in our natural history studies is where they're going to go. I mean, I grew up in the middle of nowhere, central Illinois. My sister has Down syndrome. There was no way that we were going to be able to travel, thousands of miles for a natural history study or anything like that. Could we have made it to Chicago or St. Louis? Yeah, that was probably something that could have happened for us. So I think getting more centers involved and creating a network, and then we have that network to leverage to when we do have therapies to try. And we talked about this last year too, and I think we have come a long way. I mean, I already know so many more people from different institutions, so I think that network is being built. And then to your point about what we're doing next is, creating a place for all of the data that we all have to come together so that we can all use it. And I think that's what's next.
Yeah, and let's pivot because I want to keep us on schedule. So, the 12-month question, we have made good progress. For those that aren't familiar with the work that many of us on this panel have done together, is really just organize the data that's out there to understand what fields are being collected, how we can centralize things, how we can make sure that families get access to the data. Those are some of the things we've been kind of working on. It's been challenging, I'm not going to lie. I did not think it would take us a year and we wouldn't be done. I thought we'd be presenting abstracts and posters, but I understand those issues. But what are we going to do in the next 12 months, and how then that can inform getting these treatments and therapies developed and then to the kids who need them? Zach, I want to start with you. Well, only because you've done a really nice job with the Ravicti, and I know we're going to talk about small molecules in a minute. But you moved it quickly, quicker than most could have. What are we going to do in the next 12 months?
You're very nice. So yes, I will get my paper published. So I think there's a lot of stuff moving from our end. I think the EEG biomarker stuff is very exciting. The seizure data, to try to get that sort of peer-reviewed is exciting now that we have all 50 kids. I think I would really just amplify, we're all working in silos even though we're friends, and we need to get a little closer and figure out how to do this. Pam, I was, as you were giving your talk, I was thinking like, oh, I got to get her EEG data to Jacob, and like, how are we going to do that? How many data use agreements do we need to sign? And Marie, with your detailed natural history work and thinking about what's going on with UCB, like how are we going to interact and make sure that this information carries forward, and that the collection of what we're knowing, of what we learned together, will go forward. So this conference that Amber throws is ultimately the best engine for that, because we hear stories from you as the families, and it's like, oh, we need to work harder. Families, please know that your voices are being heard. As for specifics, I mean, we have a meeting on the books for this week, so we're going to do it.
I was just going to say, I have lots of plans. I just need someone who can make them happen.
Well, and I think one of the other things I'll add to this conversation is, if we're going to develop products or treatments, we need to agree with what outcomes and how to collect those outcomes. And I think that's part of our struggle is, there isn't a great test for this specific, or how do you, like we talked about earlier, measure the number of seizures? Do you video your child every day during the study? What are the outcome measures? I mean, Yohi and I have talked about this for now almost 2 years, right? Because it's not an easy thing, but that's where I feel we need the voices of what you guys said. And so one of the things I want you to talk more about is, how can we use the work that you've already started and you presented today to make sure we're all collecting important information for outcomes?
Yeah, so in doing the study and pulling together all the data, like I pointed out in my presentation, new things came up, like things that we never thought to ask about. It was just, these things came out while we were going through charts. for things like the eye flutters, and do they eventually turn, how often do they turn into seizures? How often are they captured on EEG, not seizure, and then later captured on EEG, are seizure? So those are questions that now we know to ask those. We can create, like, I envision like a master spreadsheet that we have the questions we want answered, laid out, that someone can just enter in a way that's easily understood and accessed. What was the second question?
No, that was mostly it, just around that. And then Scott, go ahead.
I was just going to say, I mean, a big part of what we're talking with novel therapies, whether it's a gene transfer or gene editing in the not likely too distant future, and some already in non-neurologic conditions, ASOs, is this concept of platform technology, where things you learn from one scenario allows you to translate to doing that faster or better in another scenario. If you're talking about gene transfer therapy, you've got a capsid, you've got a promoter, you've got a gene. You may be able to use the same promoter, same capsid in another gene pretty quickly, and then it becomes plug and play. Maybe that's a little generous, but that's the idea of where we're trying to go, so we can bring therapies even to rare diseases much faster. What we have not done a great job of is applying those same principles to the clinical characterization of patients and clinical trial readiness. And we can have platform approaches across diseases that are rare. We've spent a lot of time talking about all the things that are unique about SLC6A1, but there's a whole lot of things about it that are actually very similar across other neurodevelopmental disorders and DEEs. And if we can capture those in other ones, we can capture those in SLC6A1. And then we just have to do this much extra effort to capture the unique thing, because 80 or 90% of it is all part of the platform, the master protocol that allows us to continually do better and better. And then when the therapy is there, you actually already have all the right information to be able to jump off, and you have the characterization, you have the lead-in data from those populations. And I hate to say this, guys, but the first therapy isn't going to be perfect. Cure means you don't have disease anymore. That's not what's on the table with any of our therapies right now. We hope it's meaningful improvement in the right direction, and as soon as we get that, we have a new natural history, and we should be working on version 2.0, the next treatment that's going to make it even better. And so this is an ongoing thing, and we need to set up clinical platforms of supportive, multi-center studies that allow us to aggregate across rare diseases rather than split across rare diseases.
Yeah, and I'll end because I know we're at extra time. But the one thing I think, again, I'll come back to, and during presentations, I always repeat things that I think are super important. That's a big thing I always do. And again, Zach knows all my tricks. That's why we have to come together and agree on what we're going to measure, how we're going to measure it, and do it the same way. Zach and I have railed for years on how we document seizure frequency and agreeing to do that so we can actually capture all that information. Because that foundation has to be set for when Allison comes up with the next big thing, right? Allison is working her tail off, and all those other PhD scientists in here. When they're ready to come to us, we have to have that foundation and be ready to know what we're going to follow, how we're going to follow it, when we're going to follow it. And that is largely going to be because of the work you've done and continue to do. So I think that's really what I would like to see us finalize in the next 12 months.
Can I do a quick plug?
Yes.
I've seen Zach, I remember. So I'm also in the ELIS group, the Epilepsy Health Learning System. So thinking of standardizing our language, these are smart forms that we use in our all our epilepsy and genetics patients that basically force you to classify the seizures, classify the epilepsy, list etiology, pretty much everything that you would want to know, including things like associated conditions, developmental, psychiatric, etc. And it's just a matter of, like right now at CHOP, most of us use it, and when we do our data mining, it's very easy. You can pull it and you have the same language as opposed to someone just writing big seizure, the little seizure, mini absent, things that we're like, what are we doing with that? And so if we can do that across and agree on that, and I know, Zach, you work on that a lot. So plug for that moving forward, because that will allow us to also all be on the same page about what we call what and what we are looking at in all areas.
Well said. All right, I know we can do this forever, but I know, Amber, we have a lot of other things that have to be covered. But I really want to say thank you again for, Amber, allowing me to moderate this session and be a part of this wonderful group. And to thank you guys for all the work we get to do together. It's really meaningful and fun, and we have a lot more to do, as you could tell.
We'll try to do better next year.